AppSec's online support program, Class 9, Week 13, Lesson 16, Subject Biology. Textbook Biology, page numbers 161 to 162, Unit 8, Nutrition, topic Small Intestine, resources required, textbook Biology. Objectives. By the end of the lesson, students will be able to revise small intestine, parts of small intestine, duodenum, jejunum and ileum. Function of villi. Small intestine, complete digestion and absorption. Now, when the food leads to the small intestine from the stomach, so complete digestion and absorption of the food will take place. Small intestine itself consists of three parts: duodenum, jejunum and ileum. We will be discussing each and every type in detail. Duodenum, the first part of the small intestine, comprises of the first 10 inches, that is 25 centimeter of small intestine, and it is the part of the alimentary canal where most of the digestive processes occur. Now, duodenum is 10 inches long or 25 centimeter long of all of the small intestine. And here it is that part where most of the digestive processes occur. Why? Because here three different secretions mixed with the food material. And that secretions come from the bile, from the pancreas and from the intestine, from the intestinal wall. Liver secretes the bile, pancreas secretes the pancreatic juice, and intestinal wall secretes the intestinal juices. These three secretions will mix with the food and they will help in the digestion of proteins, carbohydrates and lipids. Functions of secretions. Now first we will be discussing bile. As you all know, bile is the secretion of the liver. When liver secretes bile, so it comes to the small intestine and it helps in the digestion of the lipids. How lipids are digested? Lipids are digested by the process of emulsification. What is emulsification? Emulsification is that process in which the lipids are kept in the droplets form and they are kept separated from one another. Now, pancreatic juice. From pancreas, pancreatic juice is released, and itself pancreatic juice consists of three enzymes, that is trypsin, pancreatic amylase and lipase. Trypsin act on the proteins and it digests the proteins. Pancreatic amylase act on carbohydrates and digest the carbohydrates. Lipase act on lipids and it digests the lipids. Now the third one is the intestinal juices. Intestinal juices are actually secreted from the wall of the intestine, contain enzymes for the complete digestion of all kind of food. All kind of food means carbohydrates, lipids or proteins. Right, students? You can see the diagram. You can see the stomach. Just above the stomach you can see the liver, and liver you can see the gallbladder in green color. So from stomach starts the small intestine. The first part of the small intestine is the duodenum, as it is shown on the right side. So then you can see the jejunum, that is the second part of the small intestine, and then you can see the ileum as well, that is the last part of the small intestine. Jejunum, the second part of the small intestine. It is about 2.4 meters long. It is concerned with rest of the digestion of proteins, carbohydrates, lipids of our bite. Okay, now as we have discussed in the duodenum that three different enzymes lead the duodenum part of the small intestine and it helps in the digestion of the carbohydrates, proteins and lipid. But if any of these things are remain undigested, so they are being digested by the jejunum. Ileum, the last part of the small intestine. It is 3.5 meter long. It is concerned with the absorption of the digested food. Up to duodenum, up to jejunum, I have told you that digestion take place. But in ileum, absorption of the digested food take place. The food that has been digested, the proteins, the carbohydrates, the lipids that have been digested. So now the absorption will be done by the ileum. These what actually ileum are. They are circular folds in the inner wall of the ileum. There are circular folds. They are called the ileum. These folds have numerous finger-like projections called villi. Now, inside the ileum, finger-like or thread-like projections will be present, and they are known as villi. What villi actually help in? Villi just increase the surface area of the inner walls, and it helps a lot in absorption of the digested food. The main function of the villi is in the absorption of the digested food. Small intestine. Now we will be discussing the mechanism of the villi in detail. So each villi is richly, just have a look on the structure of this. We are going to discuss the villi structure. Villi is richly supplied with the blood capillaries and vessels of lymphatic systems called lacteals. Now villi consist of two things, that are blood vessels and the lymphatic system, and it is also called as lacteal. The villi are single cell thick. The digested molecules, that is simple sugars and amino acids, are absorbed from the intestine into the blood capillaries present in the villi. As we have discussed, that digestion takes place inside the small intestine. Which kind of digestion? The digestion of the simple sugar and amino acid. When they are digested, so then they will be absorbed into the small intestine in the part of the villi. But then they will come towards the blood capillaries, right? As I have told you that villi consist of the blood capillaries. So the absorbed food will come towards the blood capillaries. Blood carries them away from the small intestine with the hepatic portal vein and goes to the liver for filtering. Now all of the molecules have been absorbed into the blood capillaries, but from the blood capillaries it will goes to the liver with the help of the hepatic portal vein. Why it goes to the liver? It goes to the liver because here filtration take place. The molecules that has been absorbed into the blood capillaries, first it has to be filtered. So it goes towards the liver with the help of a vein that is called the hepatic portal vein. Now when it reaches the liver, so all of the toxin, all of the extra material will be removed and the extra food will be stored on the liver. From liver, the required food molecule goes towards the heart with the hepatic vein. Now, as we have discussed, that toxin will be removed. As the toxin is removed, so the food will goes towards the heart with the help of the hepatic vein. When it reaches the heart, so what will happen, that the fatty acid and the glycerol present in the small intestine are absorbed into the lacteal of the villi. Now we have discussed two things in villi, that villi consist of blood capillaries and the lacteals or the lymphatic ducts. As the food has been absorbed, it has been filtered, so what will happen, that the fatty acid and the glycerol will be absorbed into the lacteals of the villi. Lacteals, what will happen after that, lacteals carry them to the main lymphatic duct from where they enter into the bloodstream. So when the whole process is done, so then the food, the pure food or the pure molecules, that are fatty acid and glycerol, they will come towards the bloodstream with the help of the main lymphatic duct, and from here it is distributed in all part of the body. You can see the structure here. You can see the structure of the villi. So you can see the hepatic portal vein, you can see the liver, you can see the muscle layer, you can see even the finger-like projection that are villi. Right, students? You can see the villi, you can see the lumen. So students, this was your today's lecture. Hope you find it easy and interesting. Thank you so much.